Welcome to the Bio 341 lecture on natural selection. Of all the four forces of evolution, genetic drift, gene flow, mutations, and natural selection, only natural selection provides an adaptive mechanism, and this is the force that Charles Darwin was focused on when he wrote Origin of Species. And his four postulates, as we've reviewed in previous lectures, individual variation exists, variation is heritable, some individuals are more successful at surviving and reproducing than others, and survival and reproduction of individuals is not random, it is based on heritable traits. These are the ingredients that are necessary for natural selection to occur. And when you have differential fitness, differential survival and reproduction, it leads to adaptation of the population. When we talk about natural selection, we think of two different types. The first is viability selection, and this is the simplest type of selection, the easiest to measure in the field. This is simply fitness that is measured by differences in rates of survival to reproductive age. So we're not measuring actual reproductive output, we're simply measuring survival. For fecundity selection, we're actually talking about differences in reproductive output. This is a much more realistic measurement of fitness, but usually much more difficult to measure in the field. But the important point is that natural selection is all about who lives and reproduces and who dies and who doesn't reproduce. And the important factor here is fitness. So we'll be looking at models of natural selection in the classroom using Avita. But the basic idea with natural selection is simply that you have a new mutation that arises that happens to be beneficial in the environment in which it arises, and selection favors it. It reproduces differentially better than the other genotypes that exist in the population, and eventually the variant becomes fixed in the population. So remember that natural selection is actually occurring at the level of the individual, and it is occurring to the phenotype. The phenotype is the physical expression of the genotype. Selection acts on the individual phenotype, but natural selection affects individuals, but remember that it is populations that are the units of evolution. So even though natural selection is killing off or preferentially allowing to survive certain individuals, it is the populations that change their allele frequencies, which is the definition of evolution. So one good example is selection in Paramyscus mice. We will be reading a paper about this soon. And Dr. Hopi Hoekstra was able to find an exact molecular mechanism by which mice were selected for white fur. Mice that lived in beach populations were selected to have white fur. And if we look at a computer simulation of the system, we might have dark-colored mice living on a dark background, dark environment, light-colored mice living in a beach light-colored environment, and we could introduce a mutation into the dark-colored environment by which we have several light-colored individuals. Now, if we add a predator to this environment, the predator is going to take out the light-colored mice preferentially and remove those mice from the population, thereby changing the allele frequencies in this population back to 100% dark. And remember that selection can act on two different types of genetic characters. One is the discrete genetic characters. Those are the traits that mutationists were looking at, such as Mendel's smooth versus wrinkled peas, or else being a carrier or a homozygote for a genetic disorder such as Tay-Sachs disease, or sickle cell anemia. Another type of trait would be a continuous trait, and those are most of the traits that we see in nature. Those are the traits that evolutionists were focused on, such as beak shape, body size, hair color, etc. These traits are called quantitative traits and are usually coded for by many, many genes. So a discrete trait will just basically be one or the other, or sometimes you might have a homozygote, a heterozygote, and another homozygote. A continuous trait will be a smooth Bell distribution. And as we've already discussed in previous lectures, a smooth Bell distribution can be created if you have numerous genes that underlie a single trait. So in this case, we have 3 different genes that underlie color of the seed. And you can actually create 27 unique phenotypes from these 3 different genes. And those 27 unique phenotypes will basically look as if there was continuous variation in the population. In this lecture, I will start by giving an example for a discrete trait, and then show you some examples for continuous traits. So, selection is all about fitness, and we actually have a mathematical definition for fitness that we define as omega. We can think of fitness in terms of Darwinian fitness, so that's the number of gene copies that a genotype or phenotype places into the next generation. So, for example, genotype A1 leaves 10 offspring, A1A2 leaves 10 offspring, and A2A2 leaves 8 offspring. So that would be the Darwinian fitness. But much easier to think about is relative fitness. Relative fitness is simply a phenotype's Darwinian fitness relative to the other phenotypes. So we always set the most successful phenotype to 1 and relate all other phenotypes to that value. So, for example, in this example from above, the fitness of A1 omega is 1, the fitness of A1A2 is 1, and the fitness of A2A2 is 8 out of 10, or 0.8. As I said earlier, we can distinguish between viability selection versus fecundity selection. And when we're talking about viability selection, we're not talking about the number of offspring being passed to the next generation. We're simply talking about survivorship. And in this case, A1 has 100% survivorship, A1A2 has 100% survivorship, A2A2 has 80% survivorship. And when we standardize that to relative fitness, we get A1 of 1, A1A2 of 1, and A2A2 of 0.8. We can do this for a slightly more complex situation where we have A1 with 70% survivorship, A1A2 with 70% survivorship, A2A2 with 60% survivorship, and in relative terms, we always standardize against the most successful genotype, in which case that is A1 and A1A2. We set those to 1, and we standardize against the 60% survivorship against that and get a relative fitness of 0.86. We can also think in terms of the strength of selection rather than the fitness, and this is sort of the glass half empty approach. So selection is just the opposite of fitness. It's 1 minus omega, and if we have fitnesses of 1, 1, and 0.8, and fitness is talking about the number of organisms that reproduced or the number of organisms that survived, selection, the selective coefficient, is talking about the number of organisms that were selected against. So the selective coefficient for A1A1 is 0, for A1A2 it's also 0, and for A2A2 the selective coefficient would be 0.2. And we can think about selection in terms of a life cycle. So we have a population with allele frequencies of B1 0.6, B2 0.4. We have zygotes created with under Hardy-Weinberg expectations, 36, 48, 16. Those zygotes grow up and undergo selection. So in this case, we're doing viability selection. So 75% of the heterozygotes live, and only 50% of the homozygous B2 live. And we take that around, and allele frequencies in the population change, and they change such that B1 increases and B2 decreases. And if we take that and think in terms of dominant versus recessive alleles, we can also predict how selection will affect a population with a dominant allele that is being selected against versus how a recessive allele is being selected against. In this case, we would say the fitness of A1 is 1, and because it's a dominant trait that's being selected against, the fitness of A1A2 would be 1 minus S, as well as the homozygote would be 1 minus S. For a recessive trait, it would be just the opposite, where the recessive trait is being selected. The only time that selection sees that trait is when it's in the homozygous recessive individual. And in that case, the homozygous recessive individual gets 1 minus S. The heterozygote has the same phenotype as the A1 homozygote, and so they don't see any effect of selection. So we can simulate this in our simulator, allele A1. If we want to simulate selection against a dominant trait, we can set the fitness of A1 to 0.8 and the fitness of A1 to 0.9 and run that. And we see that against a dominant trait, the trait is almost immediately eliminated from the population within about 50 generations. That's very quick in evolutionary timescales. If we want to try selection against a recessive trait, then we set the heterozygote back to 1 because the heterozygote is expressing the dominant trait, not the recessive trait. And we can try looking at that, and you see that for a recessive trait, it takes a lot longer to be removed from the population. And this is why there are so many recessive genetic diseases that hang out in the human population. It's very difficult to remove them because they can hang out in the heterozygote and be masked from selection. So what about a new mutation that has just entered the population? Let's say we have a population of 100, 50 individuals, each with 2 alleles, so 100 alleles in the population, and a new mutation enters, so it'll have a frequency of 0.01. 1 out of 100 alleles will have this mutation. And let's say it's a very beneficial mutation, so that the fitness of, and let's say it's dominant, so the fitness of individuals that have this mutation will be 1, and fitness of individuals that do not, the A2 homozygote, will have a fitness of 0.9. We see it goes to fixation. This trait, which is dominant, will almost very, very quickly over the course of a few hundred generations go to fixation. Now what about for a recessive trait? For a recessive trait that's being selected for, that means that the heterozygote and the homozygote for A2 will both show less fitness than the homozygote A1. We can try that, and you see it takes a long time for a recessive trait to become established in the population. But once it starts to get established, then it very quickly goes to fixation and takes over the population. And just as I said earlier, we see very quick rates of fixation for dominant traits and incompletely dominant traits, but it takes a lot longer when a trait is recessive for it to become fixed. So we can make a population genetic model of selection, and it's just really based on Hardy-Weinberg. So we start off with our Hardy-Weinberg frequencies. So we have 3 genotypes. Those genotypes are expected to be in the population at p squared, 2pq, and q squared frequencies. And then we just simply add in fitness into the model. So the fitness of A1 is omega 1,1, the fitness of A1A2 is omega 1,2, and the fitness of A2 is omega 2,2. And we simply can take the product of each of these, and that will allow us to get the mean population fitness. So if we take each of these values and add them together, it will give us a value that's less than 1 because we're multiplying it by these fitness values, and that will be the mean fitness that we can estimate the frequency of the adults in the next generation. So the frequency of A1 adults will be p squared omega 1 over the mean fitness. The frequency of heterozygotes in the next generation will be 2pq times the fitness of omega 1 over the mean fitness, and the frequency of A2 adults in the next generation will be q squared omega 2 over mean omega. And here's our full population genetic model. The frequency of A1 is given by P, and P in the next generation is going to be P squared omega 1 plus PQ omega 1 notice there's no 2, over the mean fitness. And we can also calculate the change in P using this calculation. So now we're going to move on to talk about different modes of selection. And for each mode of selection, we're going to apply the population genetic model that we just discussed. So here's our population genetic model of selection. And directional selection occurs when omega for one of the two homozygotes is greater than omega for the heterozygote or the other homozygote. So omega 1 is greater than the heterozygote and omega 2. Or the opposite would be omega 1 is less than the heterozygote and omega 2. And this is just the classic selection scenario. So we have, just like we saw, we have selection occurring against the heterozygote and the homozygote A2, leading to changes in allele frequencies. And the example that we're going to use is a discrete example. We're going to talk about sickle cell anemia. So sickle cell anemia is a disease, genetic inherited disease, found in areas where humans live with malaria-bearing mosquitoes. And it affects the ability of red blood cells to carry oxygen. So we saw in an earlier lecture that the mutation to this gene, the hemoglobin gene, creates a defective hemoglobin protein that tends to become a sickle shape when in situations with low oxygen. So the healthy allele will be HA. The homozygous individual would show no effects of the disease. Heterozygotes would have one copy of the diseased allele, HS, and homozygous individuals would have HSHS. And these individuals usually die before they're able to reproduce. So we have a problem here. In a village in South Africa that has a population of 100, 36 villagers are homozygous for the healthy hemoglobin allele, 48 are heterozygous, and 16 individuals have sickle cell anemia. Sickle cell anemia kills 20% of A2 homozygotes before they can reproduce. In the absence of malaria, what are the new allele frequencies in the next generation? And what is the expected number of each genotype in the population of 100 individuals, where we've had some individuals born to replace the deceased? Okay, so the first step for us will be to figure out the fitnesses. The fitness of the healthy allele is omega 1, and they have 100% survivorship, so fitness is equal to 1. Fitness of the heterozygote is omega 1, and they also have 100% survivorship, so they have a fitness of 1.0, and the fitness of the homozygote with the disease allele is going to be omega 2, and they have only 20% of them survive, therefore their fitness is 1 minus 0.2 or 0.8. Now there are 100 individuals in the population, so they each donate 200 alleles. So our first step was to find the fitnesses. The second step is to find the number of the frequency of each allele in the population. And we do that just like we do for a Hardy-Weinberg problem. So the frequency of HA is equal to P, and that is equal to 2 times 36 plus 48. So each of the homozygotes is donating 2 alleles. Each of the heterozygote is donating 1 healthy HA allele, and that's out of a total of 200 alleles in the population. And that will give us 0.6. Q is 1 minus P, or 0.4. Next step is to figure out the mean fitness. Remember that mean fitness is P squared times each of the fitnesses. And if we plug in each of the fitnesses and plug in each of the P and Q values, we get a mean fitness for the population of 0.968. Next step is find allele frequencies in the next generation. So frequency at t plus 1 is equal to p squared omega 1 1 plus PQ omega 1 2 over mean fitness. And if we plug in the mean fitness as well as all the other values that we already have for each fitness and for P and Q in the previous generation, we get 0.62. And Q in the next generation is therefore equal to 1 minus 0.62, or 0.38. Finally, we want to find out the number of individuals that have each genotype in the next generation. That's just going to be P squared times 100 for HA, and that's going to be 38. 2PQ times 100 is going to come out to 47 heterozygous individuals, and Q squared times 100 is going to give us 14 individuals that still carry the disease allele. And notice that evolution has occurred. The allele frequencies have changed. P has gone up and Q has gone down. So that was a mathematical model for a discrete trait, a disease allele. Now we want to look at directional selection on a quantitative trait that is normally distributed. If we have selection against individuals that are on this tail of the distribution, it will end up moving the entire mean of the distribution to the right. So examples of directional selection on quantitative traits would be any kind of selection you can think of that moves a trait in a certain direction. So the wind tunnel ability that we saw in the fruit flies, or drug resistance in tuberculosis, or artificial selection for specific traits. One example would be sexual selection for certain male characteristics, for example, the male peacock. We know that in birds like peacocks, there are many males that have zero reproductive output and a few males who have a lot of reproductive output. So a few males that have over 30 plus offspring in this graph. When this occurs, and when females are able to select which males are successful, and they select based on a certain trait, you can have massive selection that removes most of the males from the population, of the breeding population, and only a few of the males with the most desirable trait will be able to reproduce, and it will move the entire distribution very far to the right. Another example would be directional selection for azidothiamidine. So if you would have mutations in the reverse transcriptase gene for HIV, that would move the selective the resistance for the entire population towards greater resistance against AZT. And finally, if you have selection, for example, for brown mice or white mice, you have selection against light fur. It'll move the population towards having darker fur. So the next type of selection I want to talk about is heterozygote advantage. So this is an equilibrium where we have selection that occurs against the homozygote for one allele, but selection also occurs against the homozygote for the other allele. And so you get what we call overdominance, in which the heterozygote is favored. So sickle cell anemia is another example of this. In the case of sickle cell anemia, we have directional selection that is moving away from the disease allele. But it turns out that in heterozygotes, the disease allele actually confers resistance to malaria, because when the hemoglobin goes sickle, when the red blood cell goes sickle, it kills off the malarial parasite. So now we have an advantage to the heterozygotes, where the homozygotes for the disease allele will still often die before reproduction, and the homozygotes for the healthy allele will be exposed to the effects of malaria and will also have it be at a selective disadvantage. So in the presence of malaria, we would expect that the heterozygote genotype. So you can see that the frequency of the sickle cell allele maps almost perfectly with the frequency of malaria in Africa. In areas where there is no malaria, we basically have directional selection, which is removing the sickle cell allele from the population. In areas where there is malaria, we find that we have overdominance. We have selection that is maintaining the allele in the population. So for our heterozygote example, in a village in Africa, there is a population of 100. 36 villagers are homozygous for the healthy hemoglobin allele, 48 are heterozygous, and 16 individuals have sickle cell anemia. And again, the sickle cell anemia kills 20% of them, but now we've added selection on the other side of the equation, where malaria is killing 30% of the A1A1 homozygotes before they can reproduce. So what will be the allele frequencies in the next generation? And what is the expected number of each genotype in the population of 100 individuals? So our first step, just like in the previous problem, is to find the fitnesses. So first step, fitness, and the fitness of the healthy allele homozygote, HA is equal to 1 minus 30% of them were dying from malaria, so it's equal to 0.7. Omega 1,2 of the heterozygote is equal to 1, and
omega 2,2 of the disease allele homozygote is equal to 1 minus 0.2, which is equal to 0.8. Second step is allele frequencies. And we happen to know already that allele frequencies are 0.6 and 0.4. Third step is to calculate mean fitness, which is omega hat equals p squared omega 1 plus 2pq omega 1 plus q squared omega 2. That ends up being equal to 0.86. That's a lower mean population fitness because we have selection occurring on both homozygotes. Now we calculate new allele frequencies. So p t plus 1. So the frequency of the healthy allele at time t plus 1 is going to be equal to p squared omega plus pq omega 1 over mean omega, and that comes out to 0.57. And then we can easily figure out that q at t plus 1 is equal to 0.43, 1 minus 0.57. So we notice in this case that the frequency of p has gone up, frequency of q has gone down. That's because there's stronger selection on the homozygote for the healthy allele than there is for the homozygote on the sickle cell allele. And then to figure out the genotype frequencies in the new population, or the numbers of individuals in the new population, we go p squared times 100 is equal to 33. 2pq times 100 is equal to 49. And q squared times 100 is equal to 18.
So if we look at a simulation of overdominance, so this is carrying that same calculation over hundreds of generations, we can have a new allele starting at a frequency of 0.01 and have selection of 0.7 or fitness of 0.7 and 0.8, just like we had in the problem. And we'll find that the allele, the disease allele will come to a certain frequency within the population and stay there. So overdominance is the only type of selection which maintains both alleles in the population. Because the heterozygote is favored, and the heterozygote has both alleles in its genotype, it maintains those alleles in the population and it reaches an equilibrium. So the continuous analog to overdominance, when we're talking about a continuous trait, is having selection on both sides of the normal curve. That tends to narrow the normal curve towards the mean value. Selection for some optimal number of offspring is an example of balancing offspring. So you can imagine birds laying eggs in nests, and if you lay too few eggs, then you don't have enough offspring and you're not very fit. If you lay too many eggs, you're not going to be able to take care of them all, and you're also not going to be very fit. So there's some intermediate value for the number of eggs that will depend on each species, but it will typically narrow to some optimal number of eggs that is laid in the nest. The third mode of selection is underdominance or heterozygote inferiority. In this case, we have selection where the fitness of the heterozygote is less than the fitnesses for the two homozygotes. And we can imagine a cross between New Zealand mice that have been diverging in different populations for a while. So some of these mice have black coats and some of them have white coats. And the allele frequencies are 0.6 and 0.4. The homozygotes are healthy. The A2 homozygotes do have some amount of disease that kills them. And the heterozygotes have a severe autoimmune disease that kills 30% of them before they can reproduce. So now we'll work through this problem.
So the first thing, as always, will be to find the fitnesses. Fitness of the A1 allele, A1 genotype. 1. Fitness of the heterozygote. %30 of them are dying, so the fitness is 0.7. And fitness of the other homozygote is equal to 1 minus 0.1, 0.9. We are given the allele frequencies in the problem, so frequency of A1 is 0.6, frequency of A2 equals 0.4. Calculate the mean fitness as omega equals p squared omega 1 plus 2pq omega 1 plus q squared omega 2 is equal to 0.84. Then the frequencies at t plus 1 going to be p squared omega 1 plus pq omega 1 over mean fitness, and that comes out to 0.63. Q at t plus 1 is equal to 1 minus p, or 0.37. So we see in this case that the frequency of A1 or the frequency of A1 has gone up and the frequency of A2 has gone down. Our final genotypes, p squared times 100 equals 40. 2pq times 100 is equal to 47, and q squared times 100 equal to 14.
So underdominance will tend to lead to this unstable equilibrium, where if the allele is at a certain frequency in the population, it will tend to go to fixation and dominate the population. Or if it is at if it's below that threshold, it'll tend to go to extinction and be lost from the population. The analogous type of selection for a continuous trait is disruptive selection. And disruptive selection occurs typically when you have two divergent lineages that have been living allopatrically, meaning in two different geographic locations and adapting to two different habitats. When those two populations interbreed, there will be selection against the hybrids, and the hybrids will basically have the mean trait. And there will be selection against those hybrids, and it will lead to a bimodal distribution. Disruptive selection can also occur due to niche partitioning, so different groups of organisms becoming adapted to using different ecological niches, or due to sexual selection. An example of disruptive selection is seen in grasses that live on contaminated soils. So a grass population that's growing in soils that are bearing heavy metals will adapt to those conditions and develop the genetic qualities that allow them to deal with those heavy metals. Grasses growing immediately next to them that are not on contaminated soils will continue to adapt to their normal soil environment. When you cross the two, you will have this trait of metal sensitivity, and you'll have, when you cross the two, there'll be selection against the hybrids, because the hybrids will not be able to survive as well as individuals that are well adapted to the contaminated soil, but they'll also not be able to survive quite as well as the individuals that are adapted to normal soil. And so they'll be selected against, and you'll end up with a bimodal distribution where you have the grasses living on the contaminated soil and the grasses living on the non-contaminated soil. And you can see that this is the type of selection that would lead to speciation. The next two types of selection are frequency dependent, which means that they depend on the frequency of the allele to determine the strength of selection in the population. The first type of frequency dependent selection is positive frequency dependent selection, where the more common an allele is in the population, the more likely it is to be selected for. An example of positive frequency-dependent selection is seen in aposematic coloration. So for example, in Hellconius butterflies, these butterflies are all poisonous. That's how they defend themselves from predators. But it doesn't help to be poisonous because by the time a predator attacks you and experiences your toxicity, you're already dead. So in addition to being poisonous, aposematic organisms also need to identify themselves as poisonous by advertising with bright colors. And when you advertise that you are poisonous, it helps if other species mates are also advertising they're poisonous in exactly the same way, so that predators get the message that your particular morph is poisonous and they will learn to avoid your particular morph. So it helps to have more morphs that are similar to you in the population. For example, in 6 different populations, the more frequent morphs had less predation attempts on them. So if we look along the bottom here, we see the frequency of each of these wing morphs in the population. So very frequent wing morphs had very few predation attempts on them. Infrequent wing morphs that the predators were not familiar with had very high levels of predation. So, and you can see that that same pattern occurs in 6 different populations, where the most frequent morph has the least number of attacks. And you take all the data together, you can see that on the x-axis we have the frequency of the particular morph in each locality, and on the y-axis we have predation. And the more frequent a morph is in the population, the less likely it is to be attacked. And positive frequency-dependent selection will also lead to an unstable equilibrium, where if you are at a certain frequency in the population, you will tend to be selected for and brought to fixation. If you're below that frequency, then you will tend to be attacked and you will tend to go extinct. Finally, negative frequency-dependent selection is also dependent on the frequency of an allele in a population, but it selects for alleles that are rare in the population. So if we look at the frequency of allele P1 in this population, when the frequency is low, the fitness is very high, and when the frequency goes up, the fitness of the allele becomes less and less. A perfect example of negative frequency-dependent selection is seen in pathogens that attack biological populations. For example, human populations will develop immunity to common variants of the flu. However, rare variants of the flu will be quite successful until they become common, at which point the human population will have developed an immunity to them. So it is always rare variants that will succeed, but once they succeed too much, their fitness will immediately drop. What is another example of negative frequency-dependent selection that we have discussed in class? Stop and think about this, and if you can't find the answer, come and ask me in class or email me. Negative frequency-dependent selection will lead to a stable equilibrium, where alleles that start at low frequencies will come up to a certain level, at which point they will no longer be selected for. Alleles that start at high frequency will be selected against until they come to a certain frequency, at which point they will be maintained in the population at that threshold frequency. And so there's a pattern with natural selection in that natural selection almost always removes diversity from populations. That's because it favors one allele over all other alleles. So it will favor directional selection, which is probably the majority of selection that we see in natural populations, will typically favor one allele and bring it to very high frequency, and thereby make all other alleles become lost from the population. So it removes that genetic diversity from the population. Similarly, underdominance will tend to cause an allele to become either fixed or lost, and positive frequency-dependent selection will cause the same outcome. There are two exceptions to this rule, one being overdominance or heterozygote advantage, in which case both alleles are selected for when they are found in the heterozygote, and that maintains both alleles in the population. Similarly, in negative frequency-dependent selection, once you get to a certain allele frequency, you're no longer selected for, and you're maintained in the population at that frequency. And now just a few words about how scientists detect natural selection in populations. Recall that the genetic code is such that if you have changes in the first or second position, and sometimes in the third position, you'll end up with a non-synonymous change that will change the amino acid composition of the protein that gene is creating. By changing the genotype, which then changes the phenotype is then seen by natural selection, and it can be assumed that that mutation will be exposed to natural selection. So if you measure for a particular gene the non-synonymous substitution rate and compare that to the synonymous substitution rate, if the non-synonymous substitution rate is greater than 1, then there's a good chance that natural selection is occurring in that gene. These days, now that we are able to sequence entire genomes, we can actually look for selection in different genes by comparing different populations that might have different adaptive regimes. So Paul Hohenlohe and colleagues studied a fish called a three-spined stickleback, and they sampled them from 3 freshwater populations as well as 2 marine populations. So sticklebacks are actually a Euryhaline species that has a wide range of tolerance to salinity, and they can occur in both freshwater and saltwater. And they then sequenced large portions of the genome of the freshwater populations and the marine populations, and they measured difference between those genomes using a quantity called FST that we'll learn about later that measures differentiation between populations. And wherever FST was relatively high, say around 0.4, they were able to make an estimate that there was probably natural selection occurring in this part of the genome. So this is a really nifty way to scan through an entire genome and find genes that are under natural selection. So in summary, of all the forces of evolution, selection is the only one that's adaptive. Selection is what takes mutations and new rearrangements due to recombination and takes that variation and brings it to fixation in a population. It's not a random process, and it requires this genetic variation upon which it can act. And finally, there are these 5 different modes of natural selection which I want you to understand and be able to apply in class. So here are the concepts that I would like you to understand from this lecture and the learning outcomes from the syllabus.